Dr. Arif Anwar, who is from Genomics, who will present this talk today, which will be about the integration of genomics, immunology, microbiome studies to enhance the clinical diagnosis and research. Briefly, Dr. Arif, prior to his current role in Genomics, he held progressively senior positions in 5 genomics and bioinformatics companies in Silicon Valley, California, at the United States of America and the UK, and now in Singapore over a period of nearly 2 decades. He has been involved in several keyword first research projects, such as the sequencing of the first cancer genome, sequencing and assembly of the largest plant genome using next-generation sequencing, and the development of the first commercial software for NGS data. His recent research and commercial focus has been on applying high-throughput genomics and immunoproteomics approaches for modeling microbial infection and the discovery of early biomarkers for cancers. He held a PhD in molecular genetics from the University College London and a BSc in genetics from the University of London and Oxford. As I said earlier, today's seminar will be titled The Integration of Genomics, Immunology, and Microbiome Studies to Enhance Clinical Diagnosis and Research. Dr. Arif.
As stated, the title of my presentation, Integration of Genomics, Immunology, and Microbiome Studies to Enhance Clinical Diagnosis and Research. I think that's the theme for the presentation today is genomics has become, of course, very important, but what has become very clear in the last 2 or 3 years is the importance of combining other types of data towards coming up with a diagnosis or enhancing research. Just very briefly, my colleague, John Paul Johnson, he was supposed to do the corporate introduction. He's the commercial director of the company. He's half Australian, so he's also Mr. Raj as well, who works for Genomics, which is our partner in Oman. Okay, so I have only 3 or 4 slides on just giving you a background in terms of the company, and the rest of it is science. So briefly, in terms of Genomics, the company is headquartered in the UK, so in Oxford. This is the research and diagnostics facility in Oxford. You're most welcome to visit if you're ever in the UK. We also have a laboratory in Singapore, inside the Genome Building in Biopolis. And inshallah, next month from May, a new facility in Brunei, which will be open in late May, early June. And we also have a facility in Malaysia. And the plan, inshallah, is sometime this year is to open a lab and an office in the Middle East, most probably in the UAE. Just briefly, in terms of background, the chairman of the company and the founder is Sir Ed Southern. So we're very proud to work with him. He is the inventor of microarrays in the Southern Blot, and the company was founded in 1995 as a spin-out from the University of Oxford. The fundamental patents for things like microarrays, detecting sequence variations, are held by the company. In fact, are licensed out to people like Illumina and Agilent. So it's something we're very proud of. And still to this day, it's something which is leveraged. Lastly, other key partners. So we have a strong relationship in the UK, of course. So LGC Genomics is the UK's oldest biotech company. And we're helping to develop a platform, which I'll talk about very briefly, which is for ultra-high throughput genotyping. And we focus on clinical applications for that. The technology was actually developed by somebody who used to be Dr. Phil Robertson, who used to be at GlaxoSmithKline. In terms of clinical partners, we work very closely with the NHS in terms of accreditation, in terms of processes and protocols. We basically follow standards set by the NHS as the way to do things. And lastly, we have a relationship, a very strong one, with Supernon in California for non-invasive prenatal testing. Okay, that's the end of the description, and now I'd like to focus 100% on science. I was very fortunate to be invited to a conference which was held in London in December, which completely rewired my mind. There was a conference held in December 2014, which was entitled Treating Autism. And this was held in London. And my own background, I was brought up as a geneticist. I fell in love with genetics when I was actually 8 years old. I had this inspirational Irish teacher who made me fall in love with genetics when I was 8. And my whole life I've kind of thought about everything being centered on genetics. What recent evidence points to is that there are other things going on which we need to look at beyond the genetics. And I'm going to talk about that. And of course, we're all very familiar with next-gen sequencing. It's become, a very powerful technology. And some would say that maybe too much use of it, especially in research, too much reliance on the results, because there are lots of things going on which happen beyond what's happening at the DNA level. And this is what I'd like to focus upon. I'd like to talk about today is next-gen sequencing, of course, which is an important fundamental technology, but there are other things. One thing which we're very interested in is looking at the immune system. And the immune system in terms of diversity is many, many-fold higher in terms of diversity than our own genomes. And often it's something which is not really looked at in diagnostics and also in research. And part of that is because it's quite difficult to model the immune system. So I'm going to talk about something new that we've been working on in the UK, which enables you to model the immune system. Something new also is biomedical intervention. We've been working very hard in terms of linking genetics with biomedical intervention, things that can be done to actually treat things. And again, so much of what we do, and I'm presuming that gets done by geneticists all around the world, is you get to a diagnosis, but then in many cases, unfortunately, there's not much you can do to treat that child. And the diagnosis is really for the benefit of the family and for future children. But what about that child? What about that individual? So there are lots of new things coming out, which are very, very exciting, which I'll talk about, which is based on biomedical intervention, things that can be done for that patient or child to reach the maximum potential of that child. So these are all fundamental things, and I'd like to talk about some very new research that's come out, which I hope you'll find interesting. So let's look at some fundamental basic things. And something that I sometimes forget is we have to remember that a human body is a system. If you have a genetic mutation, yes, that might have a causal effect on the structure of that protein, and that might affect the pathway, a signaling pathway or an enzymatic pathway. But we have to remember that even in a single cell, there are things happening. It's a ball of liquid, and there are things interacting which go far beyond just having a single base pair mutation. So the full answer requires all of these things, and this little figure is meant to represent diagnostic yield. And in fact, we are seeing this in our own samples. We've now done over 10,000 clinical samples in the Middle East. And what we're seeing is when you do just DNA, which is looking at genomes or exomes or arrays or microbiomes or all of them, then you get part of the answer. But sometimes it's important to look at also what's happening at the RNA level. And the RNA level is complicated. It isn't just about genes, it's also about microRNAs and methylation, and how those things interplay and interact with each other. And lastly, the immune response, often ignored, but very, very important. And we've got several families that we've worked with where we we get part of the answer by looking at an exome or a genome, but then when you do methylation studies, you find a little piece of the puzzle that was missing that you didn't get from the exome sequence. And then we look at the immune response, and bingo, sometimes it makes a massive difference. And the nice thing is that when you combine the three together, then not only is it a diagnostic or a prognostic, but also it gives ideas for the physician, the clinicians, the medical professionals in terms of potential treatments. Sometimes very, very simple things. And I'm going to show you some very nice examples from real patients that we've been allowed to use. So it's a ball. It's not a linear equation. It's not mathematics where you get a mutation and you get a result. And there have been many publications talking about this, the fact that genomics and transcriptomics does not explain everything. So I'm sorry it's a bit small here on this, but this I think was Ed Meikler's paper. So Ed Meikler, of course, very famous, yes. Talking about missing heritability and strategies for finding underlying causes for complex diseases. But we find the same situation also in the Middle East, where often the mutations behind familial conditions are novel. This is a paper that was published a couple of years ago, Eric Lander, talking about something very similar. So very, very famous scientists talking about this concept of missing heritability, that not everything is explainable just by looking at genetics. And one thing that I'd like to start off with is talking about autoantibodies. And this, I think, is a very exciting area with wide-ranging implications in terms of human disease. Now, it's been well known that autoantibodies, so antibodies that are produced to your own proteins, for some reason, right, it shouldn't be happening. There should be no immune response against our own human proteins, but there is. And for example, in cancer, autoantibodies are produced as early biomarkers, often many years before there are visual symptoms. This has been well published, for example, in prostate cancer, colorectal cancer, and lots of other cancers. There is an immune response to one or more of your own proteins due to a variety of reasons. And one can think about the mechanism why that might be happening. And one theory that we've been working on, for example, is fusion transcripts. When you have cancer, within the tumor, there is a translocation event which leads to two bits. It could be an interchromosomal translocation or an intrachromosomal translocation that leads to two genes that were far away from each other. The most famous one, of course, is BCR-ABL, but there are lots of others that have been discovered, which would then elicit an immune response. Now, some new things, some new ideas, and I'd like to spend some time on this. And we've now done more than 50 research projects using an immune modeling technology that I'll show you in a minute. It isn't just about cancer. This is Professor Edgar Engelman, who's at Stanford, and he's talking about autoimmune reaction in type 2 diabetes. Everyone knows about type 1 being an autoimmune disorder. This is type 2 diabetes, and we also have our own evidence that there is an autoimmune dysfunction in individuals with type 2 diabetes, which could explain why individuals with similar BMIs, similar diet, similar genetics, but very different secondary and tertiary type 2 diabetes complications. And that could be due to an a differential autoimmune response. Autism, actually it's a subject very close to my heart because it's something that I've been working on from a research perspective for maybe 8-9 years. And if I just zoom into this abstract, there is very strong evidence that maternal autoantibodies play a role in development of autism. This has been well published. And during pregnancy, for some reason or another, which we don't all understand, there are maternal autoantibodies produced to proteins that are involved in neuronal development of the fetus. And they go on to inhibit normal brain development during pregnancy. And that might be one mechanism by which autism manifests. Another idea is, and this paper talks about this, is children with an autoantibody to a 62 kD protein showed aberrant behavior compared to children who didn't have that autoantibody. And part of this could be linked to the gut. And I'll talk about the gut in a lot of detail in a minute soon. So I've talked to you about the importance of looking at the immune system and modeling what's happening from an autoimmune response within an individual. Now, the key problem is how do you model the immune system? Because protein, unlike spotting, anybody can spot a piece of DNA on an array or try and sequence an array. It's very, very difficult and laborious and expensive to sequence proteins, and very difficult to actually analyze lots of autoantibodies in one go. And people have tried, if you can make antibody arrays where you have an array and you stick antibodies on the array. The problem with sticking antibodies on the array is that if you think about it from a biological diversity perspective, it's only allowing you a one-to-one interaction. A single antibody might react with a single protein. So it's a highly limited way of doing modeling the immune system. The second way that people try is using peptide arrays, so short fragments of protein, stick them on an array and see what happens. Now, that's also fraught with problems because if you only have a part of a protein, it's like trying to drive in a car and you say, well, today I'm just going to use the first 10% of it. It's not going to work. I mean, you can't really get to work. So a part of a protein, just a chunk of a protein, is unlikely to be functional just by itself. And it's also going to be missing lots of epitopes because it's just a part of a protein. So the ideal way is to have full-length proteins so you can maintain correct folding and function. And that's tricky to do because I'll give you something, a little embarrassing story. I only had 2 hours sleep last night, so. I finished my PhD in 1995, and I spent 4 years trying to express and fold one protein, and it didn't work very well, to be honest. I just scraped my PhD. So, folding proteins, even today, is not an easy thing to do, and to get functional proteins is very challenging. So, what we've actually done is we've actually developed a technology which was a collaboration between Oxford and Cambridge University in the UK. And this technology is based on a bacterial protein called BCCP, biotin carboxyl protein. You can see it here, this blue protein here. And what we've done is we've actually cloned and expressed thousands of full-length human proteins. And this work took about 12 years to do. And took lots of trial and error to get it to work. The fundamental aspect of this technology is that essentially it's a screen for folding. And I'll show you how that works in a minute. So what we have is we've made an array, and this array has 1,600 proteins on it, and they're all proteins related to the immune system. Inflammatory response, tyrosine autophosphorylation, serine kinases, tyrosine kinases, chemokines, cytokines, transcription factors, etc. So all things molecules and proteins that would be involved in the immune system, either in eliciting the immune system, amplification of an immune response, related to signaling in an immune system pathway. So it's designed to model what would happen to the immune system in an individual. The way it works is, very briefly, so there's the blue protein, this biotin carboxyl protein. On the bottom is an epitope for binding biotin. And when you have a streptavidin-coated surface, only a biotin-related molecule will attach to it. So we use that as a clever way to ensure that only correctly folded proteins bind to the array. Because when the protein is misfolded, the biotin epitope is internalized, and then you can't stick it on the array. So to our knowledge, and this sounds a little bit of a grand claim, I don't want to overpromise something, but to our knowledge, this is the only platform in the world where you have full length, correctly folded and functional proteins on an array. Lots of people have bits of proteins, but we know that all our proteins are correctly folded and they're all functional. We've done lots of studies, so this is looking at reproducibility, very important, of course, in science. So we took the same sample and looked at the relative fluorescence signal. And when we looked at it, the variation was 1.3%. The scale on this graph actually goes way up over there, so I just took a little bit of it. So it's a highly reproducible platform. And we compared it with other people's protein arrays, and our platform is very, very tight in terms of reproducibility. So the CV is the coefficient of variance. And you can see here each spot is spotted in quadruplicate, and it's a quantitative signal. So it's a very powerful platform. We then looked at functionality. So we screened on the array, there are actually 306 kinases. So a simple assay, we looked at the kinases on the array. Can we show that these kinases autophosphorylate on the array? So it's a simple assay. You take the array, you add ATP, you wash, and then you come in with a labeled antibody. And this is the result. So for example, with RET, which is it's a tyrosine kinase, which autophosphorylates. You can see here, when you incubate with this DMSO, you get nothing. These markers are just fiduciary control markers, everything else is dark. But then when you incubate with ATP, these 4 spots actually correspond to RET, and they fluoresce. And when you incubate with staurosporine and ATP, there is inhibition of tyrosine kinase autophosphorylation, and you don't get any fluorescence. We also looked at the same thing, EGFR. You can see EGFR, as you increase the molarity of staurosporine, you get a reduction in fluorescence, which corresponds to tyrosine kinase autophosphorylation. Similarly here, this is also TYR03, that's RET, FGFR2, very similar results. So this is a very important thing to say. So we have a platform where every single protein is correctly folded and functional, and the function maintains even on a solid surface. And lastly, the protein behaves as it would enzymatically and kinetically, which is very important. Now, how have we used this? We've used it for cancer. We've used it for cancer research and also for cancer diagnostics. Pancreatic cancer, very, very difficult to diagnose. Unfortunately, manifests by the time a patient comes in complaining of symptoms, the tumor is already very advanced. And I think mortality survival rates for pancreatic cancer, I think, are the lowest of any cancer because it's so difficult to diagnose early. We discovered 3 autoantibody biomarkers which have a sensitivity of 80% and specificity of 50%. So if you screen somebody very, very early on, a routine screen, you have a 50% chance of picking up somebody who might have pancreatic cancer. So that's a very, very powerful application of this technology. We also looked at lupus, which is an autoimmune disorder, and we found 2 proteins. Now, actually, we found many more proteins. The objective of the study, from a research perspective, but also from a diagnostic perspective, is to have the lowest number of proteins within the panel, because you want to have the lowest number that still give you the same result. Specificity here, very high, 83%. Sensitivity also very good. Colorectal cancer, similar results. And we've tried this now with lots of different things. We've tried it with cardiovascular disease. We've tried it with type 2 diabetes. We want to try it with autism. We want to try it with other things which we wouldn't imagine before would work. But I personally become a great believer in the autoimmune system as a key component of disease manifestation. So that's kind of like, I want to draw a line under that. So the autoimmune story is very important. Now, how do we link that to other things? And the next part is something that I'm very excited by, which is the microbiome. Now, it's some of the research that's come out of the microbiome, particularly the gut microbiome, is truly remarkable. And I've been to lots of conferences, I'm sure you have as well. But I was at a conference in London in December, which was a treating autism conference. And I've never seen such a response from the entire audience. The entire audience got up and applauded, not me, unfortunately, somebody else, presenting microbiome research. I've never seen that with someone where is a standing ovation like that. And let me go through some exciting things which are from that conference, which is something that we've started to do. And we've started to do this now. What we're doing for certain patients is we're doing their exome or genome, and we're also sequencing their microbiome, their gut microbiome. You can sequence the gut microbiome just like you would sequence the human genome. So some interesting facts. If we think about ourselves as humans, we have about 10 trillion cells. So roughly, 10 trillion can be, my daughter is 8 years old and she said, what is 10 trillion, what does that mean as a number? And I said, I tried to think of anything that I could correlate to 10 trillion. And 10 trillion is roughly, it's a little bit lower, roughly the GDP of the USA, roughly. $10 trillion a year. Apple plus Google plus Facebook plus all the US industries added together is roughly $10 trillion. So it's a very big number. However, in terms of all of us here, we have 10 trillion, roughly 10 trillion human cells, but we have 90 to 100 trillion bacteria living inside us, which is a remarkable difference. We're actually 90% bacteria if you count us based on our cellular content. And you think, okay, why should we have that? Everything has a reason in biology. In terms of cells, we're 90% bacteria, but in terms of proteins made within our body, we're 99% bacteria. The bacteria are producing thousands and millions of proteins every day that go off and do things. And you have to ask the question, what are they going off and doing? And what's the role of it? And why should humans have this huge population of bacteria living inside us? So what does it do? Some basic things. There's lots of things that we can't break down, and the bacteria break down certain complex proteins, particularly plant proteins, into things that we can digest. And it's estimated that 10% of your calorific intake wouldn't happen if you didn't have your gut bacteria breaking things down. So a good way to lose weight, actually, I was thinking maybe I should do that, is get rid of all the gut bacteria, then you get a 10% reduction in calorific absorption. Tuning the immune system. And this is really interesting, and I was thinking about this on the plane here. I flew in last night from the UK. And I was thinking, well, it doesn't, why, when the immune system encounters a bacteria, a pathogen, it's supposed to go out and attack it and try and kill it and get rid of it. But then we have all these bacteria living inside us. Why doesn't the immune system try to attack those bacteria? How does it know the difference between a friendly bacteria and a bad bacteria? And I don't have the answer for that, but I think it's a really interesting question. And lots of people have talked about this, that we need our gut bacteria in terms of tuning the immune system, knowing which people to shoot, and which bacteria to shoot, and which ones to ignore. And then the big question, the $100 trillion question, the role of bacterial proteins. What are they doing in our body? And I'm a Muslim, and I'm not, I mean, regardless of whether you're a Muslim or not, you know, everything should have a role, right? And they do. These things do have a role. Let me show you some data. This is from a patient, one of our patients. This, who's allowed to use this data. We sequenced this individual. This is a 6-year-old child who has autism, and speech delay, developmental delay, cognitive delay. And when you look at the gut, what the gut microbiome represented by the 7 major phyla within an individual, all of us should look like this, right? We have roughly this proportion of the different phyla in terms of bacteria. And that apparently is the ideal mix. And we should all have that kind of mixture, plus or minus a couple of %. This child is completely missing the Variation Microbiome phyla. 0, no, just not there. And lots of other people have seen this as well, is that in disease conditions, not just autism, that there is an imbalance of the ideal population of your gut microbiome. For example, in obese individuals, the gut microbiome has a lower diversity compared to non-obese individuals. Inflammatory bowel disease, some bacterial metabolites have been shown to lead to an increased risk of coronary heart disease. And you know, autism studies, they found, for example, increased amounts of Clostridium and other bacteria overrepresented. Now, this is really, really interesting. And if you look at the nerve connections between our brain and the rest of our body, I think there are 8 major pipes, major motorways in terms of nerves. 6 of them go from the gut to the brain. And the vagus nerve, you know, is a huge channel of nerves. So something very interesting is going on here. Now let me take you even a bit deeper into the hole. What has recently started in the US and Canada in particular are fecal transplants. So you take a healthy donor. It sounds disgusting, but I'm just going to talk about it.
Yes, but we read about this.
You've heard about it?
In Australia, there was a doctor who did multiple sclerosis by 50% recovery in transplanting, and then there were a huge amount of people going to Africa eating excreta of aborigines because they wanted to.
Yeah. So in Canada and the US, you can, for certain conditions, you can have a fecal transplant. And let me show you some very interesting, this was published by Burchik et al. In 2011, and they took NIH Swiss mice. And NIH Swiss mice are quite aggressive and exploratory, and they like to be, you know, there are photos of NIH Swiss mice trying to scare a cat. You know, they're not scared, they're quite scary mice. And the BALF-C mice, which are usually very meek and kind of like, people don't like to explore and just go around the cage. In this paper, which was an amazing paper, and I'm so shocked that it didn't make more news globally. It did make news globally, but it should have made huge headlines globally. Unfortunately, we live in the age where, you know, someone marrying somebody else makes more news than something that can change human health. But anyway, this was astounding research, because what they did was, and forgive me because it's some of the time we're discussing, is in this research, they did something very simple. They swapped out, they swapped the fecal content they took the fecal content from the BALF-C mouse and cleared out this NIH Swiss mouse and transplanted it into the NIH Swiss mouse, and vice versa. And do you know what happened?
Character changed.
Character.
But did they change the food also? Because maybe it's related to food.
No change in food.
And how does this acquire the gut bacteria from mother to child? It comes with
That's a
breast
really
milk?
good question. That's a really good question. I'm going to come to that in 30 seconds. So you swap the gut microbiome and you end up swapping the personality of the mice. So it's happening in mice. Can it happen in humans? Now, I want to answer your question. How do humans acquire a gut microbiome?
From environment?
Several sources. There is strong evidence, it's been published, going off track slightly, it is recommended to have normal delivery. Because during the course of delivery, the baby acquires a huge amount of its gut microbiome bacteria. Second thing is breast milk. Now, previously everybody thought breast milk was sterile. It's not sterile. If you sequence them, if you metagenome sequence breast milk, it's full of bacteria that can't be cultured. They're unculturable bacteria, but there's loads of bacteria in breast milk. So environment, what you eat, is also important. Yeah. So this is fascinating research. How can we apply this to human disease? Human fecal transplants are being done in Canada, and this somebody I'm working with. Her name is Emma Allen Verso, and Emma is an amazing person, and I'm really delighted to be working with her. Emma's doing modeling of the human gut, but also looking at doing mini clinical trials in terms of gut studies. And I'll tell you the line that she said. She was the one who got a standing ovation at this conference. And this is what she said. They were working on a cohort of geriatrics. So these were individuals in their 60s and 70s, and Mr. Standing Ovation did. So these were geriatrics in their 60s and 70s. And essentially what she did was, they were all infected. They all had chronic systemic Clostridium difficile infection. They carried out to this cohort, this is published, fecal transplant, and what they found was that the Clostridium difficile infection was eradicated. No antibiotics, permanently eradicated. But the thing that was amazing is some of these individuals had dementia. Some of these individuals had advanced dementia. And some of them, the dementia was cured. So that was a big reference to them. So it's really, really remarkable research. So you need a healthy donor. The key risk is donor issues. You have to really know the person is the donor. Right? Ideally, it's someone you're related to, you know really well, because you don't want to inherit conditions that person has and make it worse and end up with a different disease. So what we're doing right now is doing very, very deep microbiome sequencing for donors. So what we're doing right now is we sequence the donor at a very deep level, at a very high sequence coverage. It's like doing very deep metagenomic sequencing. So we try to identify the rarest bacteria, the rarest genome within the gut microbiome. And you have to sequence to very high coverage. And also the patient as well. Sequence the patient's gut microbiome at very high coverage to understand at species level and at quantitative level what is present in that individual. So that's fecal transplants. So it's actually related to the autoimmune system. The reason why gut disbalance is a problem is that when you have the wrong bacteria in your gut, or the wrong types of bacteria, then there is this so-called leaky gut, where certain proteins are being released by bad bacteria into the bloodstream, and the theory is some of them may be involved in causing these disease conditions.
There is no way to change it priorly, a 1 minute transplant?
Well, okay. there's two ways of doing it. I don't want to get too graphic. One way of doing it is you have a nasopharynx, kind of a tube down your throat, and they just pour it down your throat.
If you eat lactobacillus, you know, this always.
You can also do it the other way as well. Yeah.
Because, you know, we have a lot of diabetes, and it was demonstrated that if you resect the gut of diabetes is cured. You know,
Yeah.
You know, so maybe it's a bacteria, you know, in the gut.
I mean, the nice thing is that it's a permanent solution, apparently. Okay, so we talked about autoimmune, the role of the gut, and I want to talk about now how we use this in diagnostics. So, for example, with cancer, what we're doing with cancer is we sequence sequence cells from the tumor, and you have to take cells from all around the tumor, and maybe multiple tumors. I was very proud, I was very pleased to be part of a study which was published in 2008. The paper is Sugar-Baker et al. I'm a co-author on that paper, and it was the first cancer genome to be sequenced. And it was the mesothelioma lung cancer, asbestos-induced lung cancer. And what we found in that paper, one of the key things we found is, unfortunately, is within a single tumor, within a single individual, there were 5 different types of cancer genomes. So you can't just take one small biopsy. You have to take the biopsy from multiple places. And if there's multiple tumors, you may need to take lots of biopsies because they're all behaving differently, which is bad news for cancer diagnostics, bad news for cancer therapeutics, but unfortunately that's the case. And many people have since then published more work on the fact that cancer, breast cancer, colorectal cancer, consists of different types of tumor cells in the same patient. So it's important to do whole exome sequencing. I mean, how can what I've said be used for diagnostics in terms of cancer? So in cancer, we sequence multiple biopsies from the same patient, whole exome. We also sequence the normal exome, the exome from peripheral blood. And we also use the protein array to look at the autoimmune profile of that individual. And we do the transcriptome. Now you might think, hold on a second, this is going to be expensive to do. And it is expensive to do. But when you compare it to the cost of care for an individual, for a hospital, for a country, you know, it makes sense because it allows you to target treatment and target therapies. And actually, as a hospital and as a country, you save money.
So this is for solid tumor.
Solid tumors, yes.
And
Solid
blood
tumors.
cancer.
Blood cancers you have to do.
Right.
Yeah. The point I'm trying to make here is that and it isn't just about cost, it's also the patient. Survival rates, morbidity, mortality is drastically reduced if you have this information at hand and you use it as weapons to treat that cancer patient. So it's worth investing in doing that to target treatment. What can you use the results for? The first thing is accurate and precise diagnosis, really targeted diagnosis to a very high level of detail. And we have a panel of 82 clinical geneticists based in the US and the UK and in Belgium who are involved in interpreting the results, who sign off and interpret the results. Second key thing is targeted drug treatment. And many mutations that are found in cancer individuals are druggable, as in there are drugs that can be used to treat that individual. And some will work better, and some will have no effect. Sometimes there's no point treating with a certain drug because that patient doesn't have anything that will lead to that cancer being. They might feel like something will happen, but it will not happen. It's a waste of money and time. So this is very important, targeted drug treatment and dosage options. The last thing is chemoresponsiveness. So there are lots of genetic markers related to chemoresponsiveness. So if you can also identify that. So again, it goes back to my kind of story for today, which is combining multiple things together to give you the whole picture. It is never just about one type of data.
How many centers in UK doing that kind of service?
In the NHS, I mean, this is the thing, you see, and I think it's a good question. There's a big difference in terms of what is being rolled out in the UK and the US and in Western Europe. The NHS is very conservative, they take a long time to make decisions, funding is not as good as it should be, as it used to be 20 years ago. The UK is economically still not out of the woods. And yes, ideally everyone should do this, but in the UK, if you want to do all of this, it has to be done privately. In the US, this is being done routinely. If you go to Harvard Medical School and you have cancer, they'll do your cancer, they'll do your immune profile, they'll do your transcriptome, they'll do your 2 exomes for you. And give you a
That's if you pay
diagnosis.
for it, surely.
I'm sorry?
That's if you pay for it.
Yeah, you have to pay for it. That's the thing, you have to pay for it.
And the NHS study, 5-year study, doing this kind of investigations for 6 cancers, common cancers.
It hasn't been completed yet. It's still not going. The NHS is still playing around with next-gen sequencing, for example. So, we can find pathogenic mutations related to the condition, related to response, related to drug treatments and chemotherapy. So it's a very powerful picture. Now, let's look at developmental delay in all its forms, not just autism, but it can be familial, autosomal recessive, just developmental delay of any type. It can be intellectual delay or musculoskeletal or something else. What we're doing right now is, in fact, we're moving to doing clinical whole genome. The cost of doing clinical whole genome a year ago was 5 or 6 times the cost of doing clinical whole exome. The cost of doing clinical whole genome today compared to clinical whole exome is only 20%.
How much it is? $1,000? $2,000.
But so the power that you have is you can do a clinical whole genome for 3,000 or 4,000 euros.
1,000 euros.
Yeah, 3,000 or 4,000 euros.
3 euros.
Yeah. Exome is about 3 something, right? So
these are
cheaper.
latest prices?
Yeah, it's cheaper to do. It's almost as cost effective to do a whole genome as it is to do an exome. And doing the whole genome, we find many cases where you do clinical exome and it's negative. So we're getting a diagnostic yield of about 45%, 50% on clinical exome. When you do clinical genome, we're getting a diagnostic yield of about 75%, 76%. So it's a massive difference. And you find things, of course, that you can't find in a clinical exome. I mean, one thing which is very important to understand is no clinical exome is complete. You know, the best one is 93, 94, 95% of all the exons. In a clinical genome, you have, in theory, 100% coverage. Microbiome, we're doing microbiomes for developmental delay, sequence the microbiome, understand what's going on, gut microbiome. And also in some cases, if there is a link to an autoimmune condition, also looking at protein grade. So, I mean, one thing which is very important, and particularly in this region, of course, is the higher rate of consanguinity, which causes autosomal recessive disorders, of course, to coalesce, increasing incidence within the population. Now, I talked about this last time, but what we're finding is the many, not all, many of the findings where we have a positive diagnosis are not known mutations. They're not known pathogenic mutations. They're all VUS, variants of uncertain significance. And what we find is that many mutations tend to be tribe-specific or family-specific. They run only in that family. They're not present in. Sometimes we can look at a surname and predict the result and say, okay, we think it's going to be that mutation based on the family name.
We do the same. We have same.
Yeah. Now, what we've done is, and we'd like to make this available to Royal Hospital for free after Ramadan, inshallah, from September. We're trying to finish the development. We have a database called Galaxy, and we'd like to give this to Royal Hospital access to. And Galaxy basically is a database. So we have a database of now more than 11,000. So only patients who've given us consent to use their frequency data. So it's basically a database. 99% of the samples are Arab samples from this region. And we think we've done the largest number of samples compared to any one or any company. And in the database, it's basically, it's an allele frequency database. You can see here the Galaxy allele frequency. And when you get access to this software, it's just an online access. When you hover your mouse over the world map, we've split the region into the Middle East, which is that there, and we've been careful what we classify as the Middle East. We've gone all the way west to Morocco, and as north as Syria, and as far east as Iraq, not including Iran.
But so many of these people from living in UK as well. So do they have their identity?
No, in Galaxy we've only put in, as I said, Asian samples, Middle Eastern samples.
Right. But you have so many Middle Eastern people living in all over Europe. So those ones will come as Europeans?
No. We do it based on ethnicity. If you are Arab ethnicity, then you count. Okay. We also have data from all the Caucasian samples as well. So we have all the ClinVar data and the NCBI data, all of this experience. So what's the use of this? It's very powerful because you can use the allele frequency to figure out this problem here, VUSs. So if you have a VUS, and you think, okay, it's in a gene that has not been associated with a human disease before, or it's a mutation, a variant, which is in a known disease gene, but it's a novel variant. What else can you do to help? Because every time you do an exome, you get 300,000 variants. It's bloody hard to work out what's going on, which is the cause of the mutation. If you do a genome, you get 3 million variants, at least. So the VUS plus Galaxy, we use Galaxy, and you can use Galaxy from September free, to try and interpret based on allele frequency. So if the allele frequency is above 1%, and we find the same variant in 2 children with different conditions from Oman and from Qatar, then most likely it's not a pathogenic mutation. It's not the mutation that you should focus studies on. But if the allele frequency is less than 1%, and we find 2 children with similar conditions, then maybe it is a pathogenic mutation. So this is just a snapshot comparing our services. Next, we use the Galaxy database, we use the Illumina sequencer, and everybody uses the Illumina sequencer apart from Centogene. Centogene uses the ion torrent sequencer. Just one word, ion torrent sequencer. Great at calling SNPs, not so good at calling indels. Misses large numbers of indels. The other thing that we're doing is biomedical intervention. So there are a whole panel of actionable variants. We call actionable variants. So variants associated with food intolerance, detoxification, etc., etc. One thing which is very interesting, and we found it to be highly common, is mutations. This mutation here, RS731236. It's a double homozygous mutation in the vitamin D receptor gene. And I would bet that 50% of people in this room are homozygous or heterozygous for that mutation. I'm homozygous for EDR. I didn't know it. And I did my own test a few years ago just for fun. And I discovered that I was homozygous for it. I tested the rest of my family, and I found out all my children have it. And we tested their blood, and we found out they have very low levels of vitamin D. And when we looked at it across our entire database, the frequency in Asia and the Middle East is 27%. DDR, RS73172236, homozygous. And VDR, I would say, please go and test yourself and your children, because vitamin D receptor is not just about bones and calcification, but also it acts as a gene switch.
Was it because of selection? It's lower?
It could be, exactly, you're right, you're absolutely right, that's right. My son asked me the exact same question, why do we have it? I'm like, because it's selectively good. It would have been good to have it if we lived outside and we're farming, then it'd be fine. But modern lifestyle, I mean, none of us go out presumably to do farming outside or spend 10 hours in the sun every day. Yes, you're absolutely right. It's a selective marker, selective mutation, which would have been beneficial 500 years ago or 200 years ago. But in 2015, it's a bad mutation to have. So please, have yourself. And it's been linked to cardiovascular disease, it's been linked to type 2 diabetes, it's been linked to autism, it's been linked to all sorts of things. Have you had any video on that time? No. So yeah, really worth doing. So I'm just coming to the end of my presentation. Lastly, prenatal, and I think this is where it gets really exciting. The true promise of genetics and genomics lies here. The potential to, I very rarely read my stuff, but I will again. The potential to eradicate familial conditions or even conditions at a national level. As you know, the UAE is trying to do thalassemia eradication by 2020. I think they're going to get there. And it doesn't take a lot to implement this. When you think about disease burden on a nation, if you truly implement on a national level widescreen genetic screening, not diagnosis, screening, then it is, eradication is a very strong word, I don't want to say it, but lowering of inherited diseases is achievable in a very short time frame. And the UAE is doing it for thalassemia, and we're talking to the UAE government and saying, look, the GDA, and saying, look, you could really eradicate many of the things if you really put them into it and put resources behind it.
We're talking to our government about the same.
And the potential is enormous.
Do we have to do the whole genome? I mean, your idea is really thought of it. But do we have to do whole genome or a panel of specific inherited disorders?
Okay, good question. Yeah, very good question. I think to start off with, in fact, I'm going to show you my next slide. What we're planning is a panel. Carrier screening across the most common autosomal recessive conditions, plus thalassemia, and that covers 80 to 90% of many of the conditions. So I think you're absolutely right. You know, if it's screening, if it's diagnosis, yes, you might have to do whole genome. But if it's national level screening, no country can afford to do whole genome of the entire population. So you could do, and this is what we're doing already, is doing panels. A panel of a small number of known autosomal recessive disorders, including thalassemia, for example. We're also doing PGD, and I won't talk about the other things. This is PGD, very powerful technology, very powerful approach. Can be used for screening out conditions, also of course for couples that can't conceive. And one example of screening is thalassemia. Now, what we've developed is a very low cost thalassemia test. And what we've done is, and I'm sure it's the case in Oman as well, when we talk to the Emiratis or the Kuwaitis or the Saudis, they say, you know, our population is very mixed ethnically, even though we might all look similar. My great-grandmother was from Iraq, and my grandfather was from Greece, and I maybe have a relative in other ethnicities. So genetic thalassemia screening works to an extent if you focus only on, if you're Greek and you only do Greek mutations, you're going to pick up a certain level of diagnostic yield. But the only way to get above 95, 96% is to cover all the major thalassemia mutations. That's the only way. Otherwise you miss too many people. You get too many false negatives. So what we've developed is, and Charles, we're going to release this year, maybe after Ramadan, that's the plan. Everything is after Ramadan.
Similar things also in existing fibrosis.
Yeah,
so different.
Yeah, absolutely. And this test has 232 mutations. On 4 or 5, 232. Now anybody can do 232 mutations if you spend enough time and money to do that. It contains all the major, not everything at 100%, but all the major mutations for alpha, beta, delta, gamma variants for multiple ethnicities, including Arab groups, and G6PD deficiency. Now, as I said, anybody can do that, given enough time and money. The point is, the only way this works is if you do it on a mass scale. So we have developed a technology which we have developed, a different technology for genotyping, which allows us to do this at very, very low cost.
Because of the large volume of samples.
No, nothing to do with
So
that.
what will be the cost? Because we're doing it manually.
So, I mean, the cost is less than $1 per genotype, per sample per genotype. That's per genotype. If you have hundreds of genotypes, it's potentially hundreds of dollars, but if you brought it right down to a few dollars.
Can we give you our mutation list to make sure they're there? Then we will buy it
Please.
for $1.
Yeah, please. Please
And
give us
we
your
will close
mutation
the
list.
lab.
Yeah, please give us your mutation list. And we're really, the other thing we can do is we've developed some specific hardware, some machinery that is used to run this. And we can actually have you run it yourself. We can install it here so you can run it. So it can either be as a service or it can be internal. You can do it yourself. And with our current setup, we can process 2 million samples a week. So it's possible to do
So in 1
the
week.
whole of Oman.
Yes,
do that.
one half
1
week
week.
we can do all
Half a
of
week, right? Yeah, it was all Oman samples. Yeah. So this is I'm ending now. So just to summarize, from a research perspective, all the same technologies can be used. And I think this is very important to understand that DNA change may not express if you see a mutation within a research application. It doesn't necessarily mean it manifests from a research perspective. The gene expression change might look very interesting in terms of looking at it if you do transcriptome sequencing, but the transcript might be unstable, it might become dysregulated due to a microRNA or a methylation event. The immune system is important to model as part of research. So in summary, from a research perspective, it's important to use orthogonal techniques, different types of techniques, to try and give you a complete answer. So what we do, and we help a great deal in terms of coming up with project design, research project design, is maybe combining all of these things, exome, genome, methylation. Methylation is very important. I didn't talk about it today, but I think it's very important. MicroRNAs and transcriptome sequencing, as well as the immune system, and then you kind of use fancy bioinformatics to try and work out what is going on. So in summary, multiple tools is very important. And I can't stress that enough. Genomics and transcriptomics are very important fundamental technologies by themselves, but it's important to think about what is happening in the system. And people talk about systems biology. I remember 15 years ago, 20 years ago, people went on and on about systems biology, trying to model the system. And back then there were more like review papers of systems biology than real papers of findings for systems biology. But now I think is the time where we have the tools to really think about how can we look at the immune system, how can we look at microbiome, how can we combine that with information coming from genomics and transcriptomics and try and really get the most accurate diagnosis or the best possible research direction for whatever research you're involved in. So with that, I'd like to end and open for any questions that anyone